Good afternoon. My name is Leeza Osipenko, and I'm here to introduce the HTA perspective in endpoints and cancer drug development. What outcomes are relevant to patients, and who actually determines this relevance? These seem to be very straightforward questions, but the complexity of the field is very high, and we need to start with taxonomy, defining the terms, what we actually mean when we talk about endpoints in cancer. So cancer drug development has been very prolific over the past decades, and currently we have hundreds and thousands of clinical trials in cancer, either run by the actual developers of the product or in clinical practice. And we see more and more dialogues about patient-relevant outcomes. It became a very popular topic, a much-needed one. And when we hear PROs, what exactly does it mean? Is it patient-relevant outcomes, or is it patient-reported outcomes? And how does health-related quality of life fits into this? So not all patient-reported outcomes are covered by the health-related quality of life. But at the same time, not all patient-relevant outcomes are reported by patients. And we need to clearly understand who collects which data for the appraisal of these products when it comes to health technology assessment. However, the good news is that collection of health-related quality of life and patient-reported outcomes has been increasing. I would like to take you through a study that we recently conducted in multiple myeloma. So we looked at all registered trials in multiple myeloma, which is 3,179. These are completed and ongoing trials. And we found that only 12% of them collected patient-reported outcomes data. And here you can see the breakdown by phase of clinical trials. And NA stands for the trials where the phase cannot be determined, which means it's most likely to be an observational study. So here is the breakdown of those 382 trials which collected patient-reported outcomes. And as you see, most data is being collected in phase 2 and phase 3. However, data collection on patient-reported outcomes is surprisingly low. When it comes to the designation of endpoints for patient-reported outcomes, it is expected that most of these are designated as secondary endpoints. However, 25% of those clinical trials which collected PROs put patient-reported outcomes as a primary endpoint. So when it comes to the use of quality of life instruments, most trials used a generic cancer quality of life instrument. And about only 27% focused on a disease-specific instrument for multiple myeloma. The fact M instrument was used only in 6 trials. So as you see, disease-specific instruments are not widely used, and generic instruments are used only in 16% of clinical trials which collect PROs. So this is the breakdown by phase. And once again, you can see that generic cancer quality of life instrument was most used, and predominantly in phase 2 and phase 3 trials. And this graph shows data collection of patient-reported outcomes over 30 years. So you can see a significant increase in overall trials, shown by blue bars, and by pink bars, you can see the increase in collection of quality of life data, patient-reported outcomes. And it is an increase, but not as a significant increase as, for example, we may wish to see. So what problems do we have in current PRO QL collection of data? First of all, it is not a standardized process. There are recommendations, there are suggestions of how this could be done in different trials, but there is no one gold standard. It might be good, it might be bad. But I think certain guidance for some diseases might be helping to move the field forward. Quality of data collection for patient-reported outcomes is a variable, and the timing and frequency also is not standardized, and a lot of times clinical trials get it wrong, and by collecting quality of life data at wrong points may skew the validity of the results. So when data on quality of life and patient-reported outcomes is collected in trials which are looking at novel drugs, usually these trials recruit very fit patients with high ECOG status, and these trials include patients who are usually 10 years younger than patients who will be treated in clinical practice. So patient-reported outcomes and QOL data collected in these registrational trials might not be representative of what we will see in actual clinical practice. And thus, we are lacking quality of life data from real-world setting. So when it comes to HTA perspective, patient-relevant outcomes are absolutely essential for the HTA process. And HTA perspective deals with reimbursement decisions. So this is the third party which decides whether technology is worth the money to pay for the benefits that it delivers. So patient-reported outcomes for the HTA purposes must be converted into utilities for inclusion into the economic models. Not all HTA agencies use economic modeling for decision-making. However, pretty much all HTA agencies look at quality of life data collection in a very, very similar way, whether they convert this into quality or not. So I'd like to take you through the concept of converting patient-reported outcomes, quality of life measures into this utility value for the economic model to understand how this is done and why it's important to collect this data in clinical trials. So for HTA purposes, we need to look at two important perspectives: how a particular treatment extends life and basically overall survival, progression-free survival, disease-free survival, or how it improves quality of life. And quality of life has many different categories. So here you have 5 levels of quality of life, 5 different domains, but it doesn't have to be limited to these 5 domains. So QALY is the measure of, it's a quality adjusted life year, which measures health as a combination of the duration of life and the health-related quality of life. And QALYs help compare the value of alternative treatments and interventions that have very different health benefits. So whether there are people who are proponents of this approach or they reject the use of QALYs, it's important to understand that QALYs are here to stay for some time, and in order to convert quality of life measures into QALYs, we need to collect this data in clinical trials. So EQ5D is one of the generic instruments used to measure preference-based health-related quality of life to estimate QALYs and economic evaluations. And this is an instrument preferred by NICE in England and Wales, but this instrument is also preferred by many other HTA agencies. And generic measures such as EQ5D are designed to assess and compare health status in patients with different diseases. They generally well validated and reliable, but they may lack sensitivity to detect small but clinically significant changes in quality of life due to treatment for specific diseases. So EQ5D can be converted using single summary index by applying a formula into a utility value, which is ranges between 0 and 1. So we need to make sure that quality of life data collected in clinical trials can be converted in this utility scale, regardless of which measures are being collected. So, and there are different types of quality of life instruments. And there are generic measures, as I just explained, the Q5D or SF36. They can be generic measures for cancer, which we've already been introduced to RTC, QLQ, C30, but there is also FACT-G, VAS-C scale, or there are disease-specific instruments, like, for example, for myeloma, we can use MI20, FACT-MM, for lung cancer, we can use FACT-L. And collection of these disease-specific measures is also very important because they give the perspective on the actual specific issues with that disease, which are not captured by generic instruments. And these measures include items that are likely to be affected by this disease or by treatment. And it's important to include disease-specific instruments in the trial as well as generic instruments. So when we only see trials which included disease-specific instruments and they did not include generic instruments, we need to use mapping. And it's good that we do have mapping algorithms to convert quality of life data into utility values, but at the same time, the use of mapping introduces more uncertainty. So it is a technical solution, but it's always good to make sure that clinical trials include both generic and disease-specific instruments. So I'd like to conclude with suggestions what needs to be done to ensure that we collect right endpoints in clinical trials to satisfy the decision makers for reimbursement decisions. So first, if we look at the development of novel cancer agents, it's very important to make sure that patient-reported outcomes are a standard for all phase 2 trials. We see more and more cancer products coming into the market which never had phase 3 trials. So an assumption that we will include PROs in our phase 3 trial might not realize. So it's a call for product developers to please make sure that your phase 2 programs include PROs. And of course, to use them in phase 3, it may by now seem like a standard, but as you have seen from the study that we conducted in multiple myeloma, many phase 3 trials did not include PROs. So if patients are being followed up after these trials, it's very important to continue collect the quality of life data in the real world setting. Timing and data collection and choice of instruments is very important. Yes, it's important to have more than one instrument. It's important to include generic and disease-specific and generic cancer instruments, but at the same time, it's important to not overburden patient and make sure the data collection is as complete as possible. So for studies which are not run by product developers, it's very important that we collect quality of life data in registries, in observational studies, in studies conducted in clinical practice. And it's very important to make sure that we collect quality of life data in various groups of patients who have comorbidities, in patients who are unable to provide self-reported data and where data need to be collected by proxies through carers, for example, or through clinicians. So it's also important to make sure that quality of life data is current, because what happens is when a technology comes for an appraisal to a health technology assessment agency, if the data was not collected in the trials, we refer to the literature to see where are the published data. And often this data might become outdated. And when new treatments come into practice, population changes, population weight may change, population age may change, comorbidities, other diseases So it's important to make sure that we keep it current in current populations undergoing current treatments. Finally, what needs to be done is methodological work. We need to standardize and compare instruments and approaches that we use for quality of data collection. We need to give more guidance to the developers of new oncology products, as well as trialists running studies in clinical practice. And we need to validate PRO instruments. Thank you for your attention, and I'm happy to receive your questions.